Hello future doctors, how are you all? Welcome back to MedSteps YouTube channel. So today we are going to discuss about in general principles. So our topic is oncogenes versus tumor suppressor genes and are like cancer genetics essentials. Okay. So before going to video, myself I am Anand Kumar Jana, final year medical student. Okay. So let's understand the basic concept about what are oncogenes. Oncogenes are mutated or overexpressed versions of normal cellular genes called proto-oncogenes. Okay, that means oncogenes are mutated, that means maybe some genetical mutation is happening, or overexpressed versions of normal cellular genes called proto-oncogenes. What is proto-oncogenes? Proto-oncogenes like normally promote cell growth and survival. When mutated becomes oncogenes, then after in oncogenes we can see uncontrolled cell proliferation, then it leads to cancer. Okay, so that's why this proto-oncogenes we have to know basic. Okay, so gain of function mutation only one allele. So this thing everything happening because of gain of function mutation, only one allele needs to be mutated to promote cancer. So what are the things we can see? We can see like dominant mutation. Okay, so next, what are tumor suppressor genes? If you are seeing here, this is the tumor suppressor genes. Tumor suppressor genes means it's like this genes is normally inhibit cell growth or promotes apoptosis. Okay, I made one video about apoptosis and necrosis. If you didn't see that video, in general principles I explained very nicely on board. So if you didn't see that, if you didn't watch that video, please go and watch that video. Okay, so what are tumor suppressor genes? These genes normally inhibit cell growth or promote apoptosis, and we can see loss of function, and because of that thing increase cancer risk. Okay, so mutation inactivates their function like loss of regulation and cancer progression. So here main thing is we can see this is loss of function mutation. Okay, and it will increase risk of cancer. Then after we can see both two alleles of tumor suppressor genes must be lost for expression of disease. So here it requires mutation in both alleles, like complete loss of suppression, and it's like recessive mutation at the cellular level. We have one mnemonic that is like oncogenes are like a stuck gas pedal, tumor suppressors are like failed brakes. Okay, this is the mnemonic for tumor suppressor genes and oncogenes. Okay, so now every gene we will discuss under gene product and associated neoplasms or associated cancer. Let's break down the top oncogenes you must have to know for your exams and clinicals and clinical practice and USMLE and like NEET PG FMGE exams. Okay, so first gene will be ALK. Okay, this ALK is like a receptor tyrosine kinase, the gene product, and we can see associated neoplasm, that is lung adenocarcinoma. Adenocarcinoma of the lung kinase, this like this we can easily remember this thing. Okay, next gene will be like BCR-ABL. It's like non-receptive tyrosine kinase. We can see mostly in CML and all. And here one important thing is, maybe in exam they will ask one chromosome about this BCR-ABL gene, that is Philadelphia chromosome, that is T922. That means Philadelphia chromosome will be like T922. Okay, so this is the chromosome. Next we can see here, that is BCL2. This BCL2 will be like gene product will be like anti-apoptotic molecule. It inhibits apoptosis, and we can see B cells lymphomas like follicular, diffuse large, and it inhibits apoptosis. Okay, next one we can see here, that is BRAF. BRAF means serine and threonine kinase. Okay, So here we can, that gene product will be like we can see that gene associated cancer will be like melanoma, papillary thyroid cancer and hairy cell leukemia. Okay. So here main thing is melanoma, non-Hodgkin lymphoma and papillary thyroid carcinoma and hairy cell leukemia is targeted by BCR-ABL inhibitors. Okay, so this is very important. Next one, c-KIT, that means that gene product will be cytokine receptor, and we can see associated cancer will be like detect gastrointestinal stromal tumor, that means GIST. So mostly this cancer, this gene detected by CD117 stain. How? CD117 stain. Next one we can see c-MYC. c-MYC means it's like transcription factor. We can see transcription factor and associated tumor cancer will be like Burkitt lymphoma. Okay, this Burkitt lymphoma. Then after you can see here that main thing will be like T. This chromosome will be like, we can see T84. Okay, MYCIGH. This is the chromosome. Next we can see here that is HER2 by NEU. Okay, or you can tell that is like ERBB2. Okay, so here that receptor tyrosine kinase, the gene product, and we can see associated carcinoma like breast and gastric cancers or carcinomas. So here the target will be like trastuzumab, that medication we can use for treating these cancers. What medication? Trastuzumab. Only I am telling about only important things. Okay, because this oncogenes we have to by heart, we can't understand these things. Okay, next one JAK2, JAK2. So that gene product will be like tyrosine kinase, and the associated carcinoma will be like myeloproliferative disorders, example like polycythemia vera. Okay, next one KRAS, KRAS, the gene product will be like GTPase, associated carcinoma like colon, lung, pancreatic cancers, and we can predict resistance to EGFR inhibitors. Next one MYCL1, it's like the gene product will be like transcription factor, associated cancers we can see like lung cancer. Okay, next one MYCN. Okay, and we can see here that is transcription factor and lung cancer. Next one MYCN, it's like gene product will be transcription factor and neuroblastoma. Next one we can say like RET, means we can say like gene product will be like receptor tyrosine kinase. Okay, so here we can see MEN2A and MEN2B and papillary thyroid carcinoma. It's like germline mutation in MEN syndrome. Okay, that mnemonic to recall MYC family, like MYC family. c-MYC like Burkitt, MYCL1 will be lung, MYCN will be like neuroblastoma. Okay, so these are the oncogenes. Now we are going to discuss about tumor suppressor genes. Okay, so now let's go through the tumor suppressor genes that act as the body's natural brakes. First one, APC. In APC we can see WNT pathway inhibitors, and it's negative regulators of beta catenin. Okay, its associated cancer will be like condition will be like colorectal cancer or associated with FAP. Okay, APC means like adenomatous polyposis coel. Okay, adenomatous polyposis coli or coli. Next one BRCA1 and BRCA2. It's a DNA repair protein. Mostly we can see in breast, ovarian and pancreatic cancer. Okay, but mostly I saw this question about breast cancer. Okay, so it's germline mutation, autosomal dominant inheritance. Next one we can see CDKN2A. In CDKN2A we can see like P16 protein and blocks G1 S the associated condition like melanoma and pancreatic cancer. Next one, DCC. DCC means deleted in colon cancer, that associated a condition like colon cancer. Next one, SMA D4 will be like DPC4, it is like deleted in pancreatic cancer and the associated condition will be like pancreatic cancer. Next one, MEN1 means menin, we can see associated condition will be multiple endocrine neoplasia 1. Next one we can see like NF1 gene, it's like neurofibromin, like RAS GTPase activation protein, we can see that in which condition we can see neurofibromatosis type 1 or KFI light spots or List nodules. Next one, NF2. In NF2 we can this gene we can see in myelin proteins like neurofibromatosis type 2 and we can see this thing like bilateral acoustic squamous squamanomas. Okay. Next one, P10 PTEN inhibits it inhibits PI3K by AKT pathway. We can see this thing in breast, prostate and endometrial cancer. Next one, RB. RB means it inhibits E2F blocks G1 and S, and you can see retinoblastoma and osteosarcoma. This these things to hit hypothesis. Next one, TP53 gene, like P53 protein activates like P21 and most cancers will be like Fraumeni syndrome like sarcoma, breast, leukemia, adrenal like CBLA. Okay. Next one, TSC1. So TSC1 and or TSC2 we can say like hamartin and tuberin tuberous sclerosis. Okay. Next one, VHL. VHL means von Hippel-Lindau disease, it inhibits HIF1A, it's like associated with hemangioblastomas, RCC or pheochromocytoma. Next one, WT1. Here we can see like urogenital development regulators, it's like transcription factors that regulates urogenital development, and Wilms tumor, that is nephroblastoma. Here this tumor suppressor genes requires loss of both copies to initiate cancer, the two hit hypothesis. Why this thing important? In clinically two hit hypothesis like seen in like RB, TP53 and BRCA. Examples like inherited on bad copy and acquire second mutation in life like cancer develops. We have a mnemonic that is Knudson's two hit applies to suppressor genes to tumor suppressors. Like Fraumeni syndrome like germline TP53 mutation, it's like early onset of sarcoma, breast, leukemia and adrenal. Okay, so mnemonic will be like SBLA cancer syndrome. S means sarcoma, B means breast, L means leukemia, D A means adrenal. Okay, so these are the main things. So in exams very important points will be like main important things like if any patient like oncogenes that one mutation and it can lead to cancer. In tumor suppressors we can see two mutations and it will lead to cancer, and no translocations like example c-MYC and BCR-ABL, and we have to match each gene with each cancer. Okay, so these are the frequent multiple questions in USMLE, NEET PG and FMGE. Always link with gene, then after product and disease. These are the things very important. Okay, so that's all for today. If this oncogenes and tumor suppressor genes, if you have any doubts you can drop me one message in comment section. Okay, if this video is helped and clarify your cancer genetics for you, if you have good idea, if this video is useful for you, like, share and subscribe. We will meet in our next general pathology series. Okay, so stay focused and stay healthy. Take care. Bye bye.